I hope you enjoyed it so far. Today we're going to spend some more time looking at specific instrument and looking into its applications. There is another session later today at 11, and you can join us to have a look into iSeq and into the applications that entail iSeq. But for this morning, I will be talking about iSeq 100 and the applications that we can do on it. As you know, the goal of Illumina is to improve human health by unlocking the genome power. And NGS has penetrated many markets, from reproductive health to oncology to infectious diseases, as we are all aware with the latest SARS-CoV-2 pandemic, but also with, in agriculture, where we are improving human health by assisting us in improving our food, which is not directly but indirectly looking at market assisted breeding and improving our food as well. So Illumina sequencing has been with us for a couple of years, and 90% of NGS sequencing that happens out there is on Illumina instruments. And this is largely because of the superior data quality that we have with the Illumina, but also with the integrated end-to-end workflow which they provide. So we have many different library preps that we can utilize to prepare our DNA to be ready to go on a sequencer. And then we have many different sequencers and the reagents that go with it, which allow us to unlock and look into many different applications. And obviously there's many different analysis pipelines which obviously help us to analyze the data and interpret it. So, like I said, there are hundreds of applications out there, and obviously we cannot look at all of them, but we thought of focusing into the major ones, the ones that are most popular and that are most known. And we also wanted to look at specific instruments like iSeq, MiniSeq, MiSeq, and NGSeq. Specifically because these instruments are in your labs or they are available throughout South Africa in the service provider labs. So today I will be talking about the iSeq. John later on will be talking about MiniSeq. And then next week, please join us when we talk about MiSeq and the NGSeq applications as well. So to start off with the iSeq, this is the smallest Illumina instrument available out there. It's very affordable price point, and it's really easy to install, use, and implement. And it's a very good instrument for labs that have a low output of data but need fast turnaround time. So what we're trying to achieve with this instrument is to bring it into every molecular biology lab and to help with the skill development and also building the capacity, without compromising, obviously, on the quality of data and having the high discovery power, and also having the data analysis on the instrument itself. So when it comes to iSeq, like I said, it's a very low capital expenditure. It's close to qPCR instrument, but it's highly reliable and it has a simplified workflow. It's easy to set up, it's customer installable, but we're also there to assist you if you need any help. And it's also really helps us out to explore so much more at a quicker turnaround time. So when we look at the other technologies that are present in the lab, obviously our qPCR and Sanger sequencing, those two labs are golden standards for what they do, and obviously they have high sensitivity and they're well-known techniques. We all worked with them, but they lack the discovery power and they don't have as much scalability as NGS has, and also we cannot look at so much resolution like we can with NGS. So with NGS we have that extended discovery power. We don't have to have any prior knowledge of the DNA we work with, therefore we can do de novo sequencing, but also it helps us to develop other assays, maybe for qPCR or even for Sanger sequencing. And it also has that high sensitivity, and we are also able to multiplex so much more data on the run, and we can look at simultaneously at the DNA and RNA as well. So just to put it into perspective for you, if we look at the, I don't want to call it a simple experiment, but if we have 500 targets and we have 8 samples, on the qPCR instrument, it will take anything about 11 runs or 3 days. But if you are trying to put that same experiment onto a iSeq, it will take obviously much less time. You will be able to prepare your libraries and sequence it and get the data in less than 1 day. And you would be able to do that in one run, which means that obviously your costs will be much less than when it comes to qPCR. Like I said, it's really a low capital cost instrument which allows us to tune into so much more and to look at many different targets at the same time. And it allows us to have that scalability from low complexity assays to the mid complexity ones. So if we want to, put it in a lab, we can use it to either look at the library quality control, to look at the smaller complexity projects, to have projects that require quick turnaround time, but we have a small batches of samples for it. But we can also use it to generate pilot data for future studies, or also to look at the loading the smaller number of samples and validate those calls for the larger panels that we want to put maybe on larger instruments such as NovaSeq. So let's look at the few key features of iSeq 100 and what is so great about it. If you guys haven't seen it before, this is what it looks like. Like I said, it's a really small instruments, about less than 16 kgs. It's really easy plug and play instrument. You can see here that it has an automated door and a reagent tray. This is the reagent cartridge that you would put in, and it's also single-handed operation with minimal touch points. The screen is touch screen, and also it's the first instrument that is actually has a Wi-Fi on it. The onboard library denaturation happens on the instrument, so there is no need to use NaOH, and some of you are aware with the hard disks that we have with the NaOH, and that if the concentration and the pH of it is not correct, then we can have a library failure or cluster generation failure. So that kind of eliminates that hurdle where we can just load our libraries and the instrument will do onboard library denaturation for you. Like any other instrument, it comes with a radio frequency ID, which enables us to have the traceability on the consumables. And also it's important to say that all the reagents are actually contained in the cartridge itself. So that means that the instrument itself has no fluidics on it, and there is no pump syringes, difficult tubes, multiple cameras or LED lights or lasers. There's nothing fancy like that in the actual system. Everything that happens, actually the sequencing happens on the cartridge itself, and the reagents never leave the cartridge itself. So that means that your library is obviously in there, all the reagents are in there, and all the wastes are also contained in that cartridge. So it just means after the end of the run, you will take that cartridge out with all the waste in it and throw it away. And because there is no fluidics on the system, it means there is no those post-run washes or maintenance washes that we need to do with some of the other older instruments. If we look at the innovation that Illumina did in order to bring us this low-cost instrument, there are a few innovations that they have done. I'm just going to mention the most important ones, like we were talking about just now. It's a very simplified cartridge. Of course, it's a single-use cartridge. They also came up with a new way of collecting the data and actually assisting the instrument to get the data, but we're also looking, because the flow cell is less complex, we had to look at a different kind of chemistry where we look at the one-dye chemistry. So because the flow cell doesn't work with lasers or fancy cameras, it's less expensive system, but it's still it's still approved. It's still proved Illumina quality, and it's still the sequencing by synthesis technology as we know it is just based on one dye, and we don't, like I said, we don't have multiple cameras. So we'll look into each of these features in more detail, and this is just an illustration to show you that that the cartridge is actually also a fluidic system. It has a syringe that will pump the reagents. It has a selector valve which will make sure that different reagents are pumped through the system and onto the flow cell during the different times of the run. And I just wanted to illustrate to you that you have a cartridge, and the flow cell will actually go into the cartridge, and the whole cartridge and the flow cell together will go into the system, and you can just start sequencing, and it will go through. It's really dark to see inside, but if one day you are able to actually see it up close and personal, you will see that there is not much to the instrument itself. This is why it's much more affordable NGS option out there. When we look at the flow cell itself, it's a patented flow cell that, so what that means is that on that glass slide we have a nanowells, and those nanowells are specific size, so predetermined specific size, and the distance between the nanowells is also well defined. So we don't overcluster the run as we would with, let's say, with a iSeq or a iSeq, which is a random flow cell. So these nanowells would have our oligos, and you will remember from before that these oligos are complementary to the adapter that are sitting on our library. So once the library is flushed through the flow cell, the library will bind to the actual flow cell onto the nanowells of the flow cell, and the XM technology will ensure that they are hybridized and that they form a bridge amplification, and that we have a clonal amplification of our library, which is then ready for sequencing. So the way that instrument then detects these clusters is that we have what we call a Cmos chip, which is a complementary metal oxide semiconductor here at the bottom. And these guys are actually just there to detect the signal that comes from the actual library that is sitting on a nanowell that is excited by the light source. Okay, so if we look at the one channel SBS chemistry, it means that we only have a single dye, and that is a fixed dye on T. G is not labeled, and then we also have a dye on A, which is actually a cleavable dye, and I explained to you just now what that means. And we also have a C that has a linker that can accept the dye. So how this one-channel chemistry actually works is that we take 2 images during our cycle. Okay, and in between those images we have intermediate chemistry step. So what that means that, once we have our bridge amplification and we are ready to start sequencing, you will remember that SBS sequencing happens once at, one nucleotide at a time. So it means that as we are incorporating a nucleotide, it will, get that dye on the nucleotide will get excited, and it will give off the signal to the Cmos chip that then it will then detect the signal. Okay, so we will have, once the base is incorporated, we will take the image, and then we will have an intermediate chemistry step where if we have A, the dye will get cleaved off. Okay, and if we have a C incorporation, the dye will actually connect to the C. So when we take a second image, we are able to look at different signals. So like I said, we take 2 images, and in between we have an intermediate step. So what that basically means, if we don't have a signal in either image 1 or 2, it means that the G was incorporated. If we have a signal in both images, it means that the T has been incorporated, because the T is the nucleotide that always has a fixed dye. If we have signal in image 1, but we don't have anything in image 2, it means that we incorporated an A. But if we incorporate the C, it means it's other way around. It means that we didn't have any signal in image 1, but we have a signal in image 2. So that is basically in a nutshell how one channel SBS chemistry work, and this is the only instrument in Illumina range that uses this chemistry to do the sequencing, of course, without compromising on Q30 score and the quality of the data. So when you look at what is available out there on, when it comes to reagents for the iSeq, actually it's only one cartridge that is available, one kit, let's call it, but depending on run configuration and the read length that you want to do, so anything from single read, either 36 base pairs or 50 base pairs, 75 base pairs. You can also obviously do a pair end where you can look at the 2 times 75 or 2 times 150 to give you 300 cycles. So based on your read length and the number of reads that flow cell has, which is obviously a fixed number of 4 million reads, you will be able to get anything from 144 million bases to 1.4 gigabases. And obviously longer it takes to more data that you need to generate, it takes longer to do it, but as you can see, it's less than 24 hours that you are able to do to get the data from the iSeq. Therefore, that's why we say this is perfect for the labs that need high turnaround time. They don't have a lot of samples because the capacity of the flow cell is not as big as, let's say, MySeq or obviously an XSeq, but it's enough out there for certain applications, and the turnaround time is really fast. So when we look at specific applications that can be done on a iSeq, I'm not going to discuss all of them, but we will zoom briefly into a few of them. If you guys have some projects that you would like to look into, please don't be shy, just drop us an email and we can arrange one-on-one meeting and discuss these with you. So obviously when it comes to iSeq, because it's a small capacity flow cell, we can do a small genome sequencing, and that will also include de novo sequencing, so novel species sequencing. We can do plasmid mitochondrial sequencing, but we can also do a targeted approach, which is actually a very good solution for resequencing. So there is a wide range of Ampliseq panels out there, and I will touch briefly on this, but we can also look at the long range PCR if you're interested, let's say at 10 kb fragments. You can, we can share these ones, prepare the library, and then sequence it on the iSeq. We can also look at the meta barcoding if you're interested in environmental samples like water and soil, or from more clinical point of view if you're interested in, let's say, bacterial composition of a gut microbiome, where you could look at 16S rRNA metagenomics, but you can also adapt this method to look at maybe yeasts and fungi with ITS region, or to look at other animal species with cytochrome oxidase. It's also perfect to do gene editing validation, to look at the targeted transcriptome approach, and also for small RNA sequencing and HLA genotyping. We will discuss small RNA sequencing and HLA genotyping later today, so please join us if you are interested in those applications. Please join us in the next session. So you will, I hope, remember from our first session when it comes to NGS workflow that obviously we need to prepare a library, and there are many different library preps out there. We briefly discussed how is cluster generation and sequencing actually happening on the iSeq itself. And then please join us on the 14th of August. We will have a dedicated session for the DNA analysis. So please don't miss that one. So when it comes to library prep and iSeq, I think the perfect fit for library preps with the iSeq would be an Xterra DNA Flex, but also an Ampliseq range of products. So when it comes to an Xterra Flex, this is the fastest Illumina prep that is available out there, and it's very flexible. and it produces reliable and consistent results. When it comes to Ampliseq, it has obviously proven high performance, and it's simple and fast amplicon workflow. So what that basically means is that we are targeting certain regions with the Ampliseq. We are amplifying only those targets that we are interested in, and then we are sequencing only those. We can also look at the targeted approach where we would create a library from the whole genome, but then we would pull out the targets that we are interested in with bioattenuated probes. And this is so-called the enrichment protocol. So there are many different ways you can do the targeted approach, but for today I will focus a little bit more on the Ampliseq. So when it comes to these two libraries, obviously NextEra Flex is ideal for whole genome sequencing, either for small bacterial or viral genomes, but also for larger genomes, because we can have anything from 1 to 500 nanograms of startup DNA. We can also work with RNA viruses, but obviously we need to convert them to cDNA, double-stranded cDNA. And as I mentioned, you can also use it for long-range PCR products. When it comes to Ampliseq, this is our targeted panel, so it works well for complex genetic diseases or for screening routine samples of cancer patients. I just want to briefly touch on NextEra Flex. Next week we will look in more detail into principles of NextEra Flex, but I just want to bring it to your attention that this is a very versatile library prep. Like I said, it has a very broad range of DNA input, but it's also very reliable and robust. And it gives us a consistent insert size, a consistent library size, which obviously helps us when it comes to uniformity of coverage and when it comes to assemblies. And this is largely because the tagmentase is sitting on the actual bead, and this allows for the saturation of the bead, which then it doesn't allow for your DNA to be over fragmented and then not have the consistent size of the library. When we talk about whole genome sequencing, obviously with the iSeq, because of its capacity of the flow cell, we will be focusing on viruses, archaea and bacteria, because those genomes are quite small, maybe a smaller protozoas or fungi as well. And these other larger genomes, obviously we can sequence them on the larger instruments such as NextSeq or NovaSeq. When it comes to NGS in microbiology, I'm sure many of you are aware that the latest pandemic, which is caused by SARS-CoV-2, has been actually the virus has been identified using NGS, and we are able to characterize it. Because of the NGS, we were able to do it so fast, and it also allows us to then develop acute PCR methods, which are heavily used now for diagnostics and screening. Okay, but we are still using the NGS for the surveillance of the virus, and we are looking at the rearrangements, mutations, maybe evolution of the virus, and of course we will be using NGS when it comes to vaccine trials, where we look at the host pathogen interaction. So these are kind of four main kind of applications that you can do with NGS when it comes to microbiology, and obviously larger organisms. If you're interested in any plant or animal genomes, you can also look at characterization and host pathogen interactions if you're looking at infectious diseases of plants and animals. When it comes to core method in microbiology, I would like to mention a whole genome sequencing. This can obviously be either de novo from culture, or it can be just a sequencing where you're looking at, maybe you're looking at the variant calling, or you're looking at the different genes, maybe resistant genes. We can also look at shotgun metagenomics. This is more applicable to your complex environmental samples, where you would look, let's say, at the water or soil samples, where you would sequence everything, all the microbial diversity that is in there, and that will include viruses, any bacteria, maybe some nematodes, So whatever is in that soil sample, you will be able to sequence. Obviously, because that sample is so much more diverse, we need so much more data, and therefore shotgun metagenomics is more suitable for a larger instrument like NextSeq. So we will discuss this in more detail next week. And if you look at 16S rRNA metagenomics, specifically the Illumina workflow is based on V3V4 region, and that is more suitable for MySeq, which can do up to 600 cycles. So we will also look into this in more detail next week, but we can also use the same principle of this method to maybe look at the different targets, like I mentioned before, maybe ITS or COX. And also we can look at the viral, like I mentioned before, we can look at the virology, and we can look at viral particles either being DNA or RNA, but we can also look at the quasi-species and the co-infections that are present. Okay. When it comes to bacterial sequencing, obviously we want to map the novel genomes and perform comparative studies, and it enables us to sequence a very low frequency and to look at the very low frequency variants and also at the genome rearrangements. And because we are able to multiplex many different samples at the same time on the iSeq or any other instrument. We can also look at a more targeted approach of bacterial sequencing. For example, if we look at TB sequencing, where we are looking for antibiotic resistant genes, we can target those specific genes and then look at how we can which actual resistant genes are present in the genome, and then we can have a more better decision in advising which antibiotics we can give to a patient, and this will help us to deal with the disease more effectively and faster. If we look at viral sequencing, like I mentioned, you can do either DNA or RNA viruses. You can also PCR your virus. It doesn't have to be directly from, let's say, a clinical sample. You would obviously prepare it with Nextera Flex. The viral sequence is so small that you can sequence it on the iSeq. So if you're working on HIV or influenza viruses or rotaviruses, or if you're maybe interested in agricultural viruses like foot and mouth disease or African swine fever, then iSeq is a perfect instrument for you. And then you can also use base to analyze the data. Obviously, when we sequence, there's always the question of how many samples can I put on the specific flow cell, and that will obviously depend on the output of that flow cell. And it will also depend on the size of your genome. Obviously, if we work with smaller genomes, then we can put more samples on, but if we are working with larger genomes, then we don't have space for so many samples. So this is just to show you that the capacity of the iSeq is 1.2 gigs, and that means if we are working with the small genomes, anything from 5 to 10 billion bases, and if we look at the average of 30x coverage, we can put anything from 1 to 8 samples. If you're looking at obviously at the smaller organisms with less coverage, you can put more samples on it. Same can be said with a targeted approach, and we'll go into that just now into a little bit more details. But obviously, if you have more targets you have and a higher coverage you want, you will be able to put less samples on. Okay, so just to tap into the Ampliseq workflow for the targeted sequencing in a little bit more depth. Like I said, this is for the targeted sequencing approach, where you just want to look at a specific number of genes that you are interested in. You're not interested in anything else. Let's say you're working with complex genetic diseases, and there are certain genes that you want to investigate and look at the variance between the different patients. Then Ampliseq is perfect for you, because you can there are already options out there, you know, off catalog, off-shelf options out for there. But basically how this works is you will have a panel which will obviously amplify the targets of your interest. It will amplify the genes that you are interested in. And then we will ligate our adapters on. Remember, our adapters allows us to bind to the flow cell and also to multiplex many different samples. And because we have 384 different indexes available with this library prep, that is how many we can do in one run. Okay, it's very flexible library prep. The input type, DNA sample type can be it can be variable, so it can be anything from DNA and RNA, but also it can be fresh or frozen, degraded, and obviously very important FFPE samples. We can also work with very low input samples, which is great if you don't have a lot of sample, or if it's a precious sample and you cannot, you cannot get a lot of material out there. So let's look at the different options that are available. Like I said, there are 12 off-the-shelf available panels. Obviously, some of them will be larger panels. BRCA panel will obviously look at the BRCA1 and 2 genes associated with breast cancer. We have a focus panel, a cancer hotspot panel, myeloid panel that's looking at different genes associated with leukemias. And then, of course, we have larger panels such as this transcriptome panel or exome panel, and obviously these larger panels will have to be done on the NextSeq, for example. There is also a community panels, which means that there's a consortium of researchers out there that worked on this panel, but is readily available for us. And if you're interested in complex diseases like dementia, you can tap into this. Or if you want to look at tumor immune system interactions, you can also look at those targeted genes. Infectious diseases like Ebola or COVID-19. We can also look at cardiovascular diseases, pulmonary, gastrointestinal, so many different diseases we can look at. What is also great about Ampliseq is that there is also possibility for a custom panels, which means that you can decide which genes you want to put into the panel and which genes you want to sequence and assess. So just to illustrate that Ampliseq is available as a library prep for many different instruments out there. We will look into iSeq just now, but if you're interested in let's say mid to larger panels, then we will have to move them onto a larger capability instrument. So when it comes to Ampliseq on the iSeq, there's 4 panels that we can look at. That is a focus panel which looks at 52 genes associated with solid cancers like breast, lung, colon cancer, and so on. We can also look at BRCA1, BRCA2 gene specifically for the breast cancer, but we can also have a panel that looks at the cancer hotspots of 50 different genes, again associated with solid tumors. And also we can look at the immune response panel that looks at the tumor immune system interaction. Like I said, the input for this library prep is very low, 1 to 10 nanograms, and you can normalize, pool, and sequence the library per end at 150 by 150. So I just want to talk a little bit more about the custom solutions that we have out there. You can reach out to us and we can help you out with designing your own panel, but you can also do it yourself through Design Studio. Design Studio is available on Illumina website. It's free of charge. You can just create a login, and it's a very intuitive and easy to use pipeline to design your own panel. So like you can see, you can have either DNA or RNA panel. There are many different solutions for targeted panels, but there is also a solution for enrichment, so for our probe-based panels. And you can also, apart from human, there's also options for different species, and this is mainly for the enrichment panels. Just to show you how it would look like, you would basically choose the genes that you are interested in, and then the system will design the panel for you. It takes it depending on you know how many genes you are interested in. It might take some time, but you will get an email saying that your panel is ready, and it will show you what is the coverage, in silico coverage of those genes, and also what is the uniformity. And here on this pie chart, you will be able to see that if you reach the capacity for the panel, which means specifically for this example, out of 500 genes that we can do with the panel, we only did 59. So there's so much more that we can get actually from that panel for the same price. So just to show, when it comes to multiplexing, obviously of different samples, I will just focus on the iSeq and just show you that obviously less amplicons we have, more samples we can put on the run. But if it becomes more complex, obviously less samples we can put, and if it's a really larger panels, then we will have to move it to, let's say, an iSeq. Just to summarize on the Ampliseq benefits, it's a very simple and fast protocol to follow. Like you saw, it doesn't require a lot of input DNA or RNA, which is really great for your precious samples. It also has a robust performance, and it has the highest percentage sequence-based quality Q30, which also has over 90% is on target and over 90% coverage uniformity. And you are able to look at down to 5% of variant allele frequency with this type of application. So just to summarize on our talk today, I just want to leave you with a take-home message on iSeq, that this is a low-cost option for you, and that it's we're targeting to put it into every molecular lab, because we really want to build that NGS skill, and we want to work on that capacity building. So it's really simple workflow that you can follow. Like you saw, it's plug and play. It's loading your library. It's inserting the flow cell into the cartridge, into the instrument, and we are ready to sequence. Like you saw with Ampliseq panels, we have a single panel, but we can do multiple targets at the same time. Obviously, we can also do whole genome sequencing of either bacteria or viruses. We can also look at the targeted approach for those bacteria. For example, like we spoke about TB, antibacterial resistance, antibiotic resistance. And so it's really ideal for every lab that has a small batch but requires a quick turnaround time. So this is in a nutshell about iSeq. If you guys have any questions or applications that you guys want to discuss, you are more than welcome to email us. If you have any questions, you are welcome to drop them in the chat, or you can unmute yourself and we can briefly discuss it. I want to thank you for your attention, and please also remember we have another session at 11 o'clock, so please join us when John discusses MySeq and applications that we can done on a MySeq. Thank you. Adrian, are there any questions? Can you help me?
Maria, so I haven't seen any. Please feel free to unmute yourself. We still have about 10 minutes. Maria can take questions. So can I maybe just start by asking, is there anyone here who has or access to an iSeq or who is planning to do work in the next few weeks or months on an iSeq. If you are here in the session, can you just maybe type a yes or just raise your hand? Okay, I see Natalie Smith is. Anyone else? And Sandy? So maybe, Natalie, do you want to share what project you're planning to run on your iSeq?
Sure. So I'm doing a targeted sequencing on our iSeq at this. I am targeting 16 genes that are involved in the low density lipoprotein cholesterol metabolism. So I'm looking at variants or looking for variants that will either cause high cholesterol or low cholesterol, specific in African populations. And I am actually using the Nextera Flex kit, and I will be starting in 2 weeks' time with Marija.
Wow, sounds fascinating indeed. Good luck with that. So you're already talking to Marija about your project design and all that, I believe.
Yes.
Yes,
Yeah.
I've got everything.
Okay,
Yeah.
awesome. Glad to hear. Sandy, do you want to share?
Yes, so I have a bunch of projects looking mostly at metabarcoding of animals. So we use the CO1 barcode region. And we have projects from fecal samples all the way to looking at zooplankton in the ocean. So at the moment, I'm not sure about whether using the iSeq will be appropriate or using a bigger machine. So that's why I'm in this session, to learn a little bit more about the two machines.
So it will depend, if I may, Sandy, it will depend on the length of your target. So sorry?
Yeah, so we design our own primers to amplify never more than about 200 base pairs. So they're short little fragments, which is why I think the iSeq may work, but I'm not sure. I'm not sure.
Yes, no, it will work. So remember, with the iSeq we can do up to 300 base pairs. Yeah. So actually 200 is perfect. It just depends, you know, what is your how many samples do you actually have, and how much do you want to multiplex?
Yeah. Perfect. Thanks.
Yes. Pleasure.
Okay, I also see Mabasa says they're currently using the iSeq for his project, but the virology department at University of Pretoria recently got an iSeq, and they hope to use that in the near future.
Yeah, you're more than welcome to contact us regarding your project, and we can help you out with either iSeq or iSeq. So anybody out there that is still unsure about their project's design of the project, what needs to be done, please feel free to reach out to us, and we can guide you through the process. I just want to thank again everybody for joining us. If there are any more questions, please feel free to drop us an email. We are available to you. I would just also like to invite you to join our next session at 11 o'clock as well.
Great. Thank you, Marija. I don't know if there's anyone else who would want to contribute or ask a question. Otherwise, thank you again for your time. Thank you, Marija, for a really great presentation. We learned so much. And yeah, see you all again, hopefully at 11 o'clock, where we'll talk you through the miniseq, the technology behind it, as well as the applications that are possible. See you shortly. Bye, everyone.
Thank you, everybody. Bye-bye. Thank you.